Hello, and welcome to the continuing story of meiosis, Meiosis 2. Meiosis 2, like meiosis 2, like meiosis 2, it is meiosis 2. Like meiosis 1 and mitosis has all the same phases, prophase, metaphase, anaphase, telophase, but now there's a 2 after them all. Okay, so let's get started. Meiosis 1, as you recall, was the reduction division. It separated the homologous pairs. Meiosis 2 is going to separate our sister chromatids. So meiosis 2 looks very similar to mitosis 1, and that's because it's exactly the same as mitosis 1. You'll see in a minute. All right, so meiosis 2, our second division, we've already had one division in meiosis 1, occurs in both of the 2 haploid cells that were produced during meiosis 1. Okay. During meiosis 2, the sister chromatids are separated in a process, again, that is virtually identical with mitosis because it is identical to mitosis. Okay? Because the cells are haploid, there's no more homologs, so they're just kind of hanging around. Okay. So meiotic prophase 2, again, normal prophase stuff. Chromosomes condense, spindle microtubules form and attach to our duplicated chromosomes. Nuclear envelope breaks down. If it formed, it may not form. So you may not have to do that. So if it reforms, you break it down. If it didn't reform, you don't have to worry about it. Okay. And again, the kinetochores of the chromatids are attached by microtubules from opposite poles because we're going to separate the chromatids. Okay. Meiotic Metaphase 2, the duplicated chromosomes line up all by themselves, perpendicular to the spindle, just as they do in mitosis, and you get a nice metaphase plate. In anaphase 2, we're going to see a picture in a minute. The chromatids release each other at the centromere, they separate, and they move to opposite poles. And then telophase 2, normal telophase stuff, you get cytokinesis, the nuclear membranes reform, and the chromosomes decondense. Okay, so now we've gone through 2 nuclear divisions, meiosis 1 and meiosis 2. So we started with 1 diploid cell, and we produced 4 haploid cells. So we started with 1 diploid cell and produced 4 haploid cells. And here it is, meiosis 2. Now again, we're starting with 2 cells, 1, 2. Those are the 2 haploid cells that we got at the end of meiosis 1. And the next picture shows everything together. So it'll make a little bit more sense. But we're continuing from meiosis 1. So we have our 2 cells, and they are, of course, haploid because we've already reduced the number of chromosomes. We only have 1 copy of each chromosome. So we've got nuclear envelope breaking down, spindle reforming. They're going to attach, spindle fibers will attach to the chromosomes and bring them to the metaphase plate. In metaphase 2, we've got our chromosomes in both cells lined up at the equator of the cell. Once they get there, they let the sister chromatids release each other and they can separate, and 1 sister chromatid goes to each pole of the cell. Okay. And then after anaphase 2, telophase 2, we've got our 2 sets of chromosomes. We get cytokinesis. There's our contractile ring. The chromosomes decondense. The spindle fibers go away. The nuclear envelope reforms, and we're left with 4 haploid cells. 1, 2, 3, 4. Voila. Okay, and if you just look at these 4 cells here, you'll notice that look in the nuclei at the chromosomes. You have 4 different cells genetically. Okay, so this one has a solid purple chromosome and a yellow chromosome with a little purple piece on it. This one has a solid yellow chromosome and a chromosome that's mostly purple with a little yellow piece on it. Okay, this one has a purple chromosome with some yellow. This one has a yellow chromosome with some purple. And this one has a solid purple and a solid yellow. Okay, so you have 4 genetically different cells. Okay, and that's the goal. Okay, we want to have genetic diversity. All right, so here's a kind of a summary of both processes, along with a comparison comparing mitosis to meiosis. Okay, so here's mitosis. All right, of course, DNA replication happens before both processes because you have to replicate, you got to make those copies in order to distribute them to the daughter cells. Okay. So up here, mitosis occurs in body cells, while meiosis only occurs in cells that produce gametes down in the gonads. All the cells are diploid, just like the parent cells, and we get 2 cells, and they are identical genetically to the parent cell and identical genetically to each other. The result in meiosis are haploid cells. We've reduced the number of chromosome. Only 1 of each homologous pair is present. And we have 4 cells, and they are all genetically different from each other. Okay, cell divisions in mitosis, only 1 division. In meiosis, there are 2 divisions. In animals, mitosis is basically for everything: development, growth, repair, maintenance, blah, blah, blah. In meiosis, the only reason you do meiosis is to produce gametes. That's it. Okay. And then again here, if you look, here's a side-by-side comparison of mitosis and up here and meiosis down here. And it's nice here, you kind of see the 2 phases of meiosis together, meiosis 1 and meiosis 2. And as you see, if you just kind of look at a comparison, mitosis and meiosis 2 look very similar. It's meiosis 1 that is the fancy part, the reduction division, where we separate the homologs. Okay, so make sure you are kind of aware. This is a great slide to have of all of these similarities and differences between the 2 processes. Okay. Now, where does meiosis happen? And I kind of alluded to it a little bit on the previous slide. It happens in gamete-producing cells. So your gametes are made in the gonads, so in the ovaries and the testes, and that's where our gamete-producing cells are. Okay. Again, meiotic division, specialized nuclear division, meiosis, 2 rounds of cytokinesis, okay, produce 4 daughter cells that are going to become gametes. The daughter cells are genetically different. This should all be reviewed from the parent cell and from each other. Okay, each daughter cell has half or haploid the genetic material of the parent cell, which was diploid. Okay, now the interesting thing is that because we're producing 2 different kinds of gametes, right? In ovaries, we get the egg, that's from the female. And in the testes, we're going to get a sperm, and that's from the male. So the process of meiosis is slightly different in males and females because we're producing different gametes. Okay, so let's have a look at that. In males, production of sperm is called spermatogenesis. So this is what's happening in the testes. Don't worry about all these kind of fancy terms on the side. Basically, just watch. We have a spermatogonium. It undergoes DNA replication, okay, to produce a spermatocyte. The spermatocyte undergoes meiosis. Meiosis 1, again, reduces the number of chromosomes. We go from 2N to N, diploid to haploid, okay? And then meiosis 2, we separate the chromatids to get our 4 haploid cells, okay? And now these haploid cells are not fully mature sperm. First of all, they're too big. They've got to swim far and they got to swim fast. So what the sperm does to become a mature sperm cell is it basically gets rid of all of its cytoplasm. And it just becomes kind of a nucleus on a stick, a nucleus with a tail. Okay? And you'll notice here, it's really just delivering DNA. It brings nothing else with it because it's the one that has to travel. Okay? And it needs to travel light. Okay? So spermatids jettison their cytoplasm, get rid of cytoplasm. Okay? And they grow a tail, and boom, now they're sperm, ready to go. And spermatogenesis produces 4 sperm from every meiotic division. In females, things are a little bit different. In females, the production, meiosis in females is called oogenesis or oogenesis, however you want to say it. I usually say oo. Okay? So kind of the same thing. We get some early mitotic divisions here, but basically it ends up producing a primary oocyte. Okay? And then this primary oocyte undergoes meiosis 1, our reduction division. So it's diploid and it's going to become haploid. Steps of meiosis are the same, separate the homologous pairs, etc., and so on. But when cytokinesis occurs, you can see right here, it is uneven cytokinesis. If you recall, our good friend the sperm got rid of all his cytoplasm. So he's not bringing any cytoplasm with him. So the egg needs to have enough cytoplasm to get the zygote through all of the early cell divisions. So there's an uneven division of the cytoplasm, uneven cytokinesis in meiosis 1 that gives merely all the cytoplasm to one cell and none of the cytoplasm to the other cell. Okay, this other cell is called a polar body. Okay, and then the oocyte continues on with the cytoplasm. Then meiosis 2, separation of the sister chromatids. Okay, this happens. Again, we get another uneven cytokinesis. Okay, and to produce an ovum. Okay, with all the cytoplasm, you get a second polar body. Sometimes this first polar body will undergo meiosis 2 and you'll get 3 polar bodies, but 2 polar bodies, 3 polar bodies, it doesn't matter. We don't use these. They get junked. We use this one, and that is what encounters the sperm and undergoes fertilization to become a zygote and eventually a new organism. Okay? So whereas spermatogenesis produces 4 haploid sperm, oogenesis produces 1 haploid egg and 2 or 3 polar bodies. Okay? So that's some details on meiosis, finished up meiosis 2, and that's it. So make sure you can compare mitosis to meiosis, familiar with all those steps, and away we go.